Okay, sorry for the third recording, guys, but the internet connection on campus doesn't seem to be that stable. So I kind of lost an internet connection there, but I believe this is the slide that I left off on. So most enzymes will end in ASE. For example, the enzyme sucrase catalyzes the hydrolysis of sucrose into glucose and fructose. Each enzyme catalyzes a specific reaction and can recognize its specific substrate among evenly closely related compounds. The active site is the region of the enzyme, often a pocket or groove that binds to the substrate. The complementary fit between the shape of the active site and the shape of the substrate is responsible for enzyme specificity. What that means is this enzyme specificity means that the substrate binding to an enzyme is very specific for that substrate. It is like a lock and key. If it's not a perfect fit, it's not going to happen. When the substrate enters the active site, the enzyme changes shape slightly, tightening around the substrate like a handshake. This induced fit results from the interactions between the chemical groups of the substrate and the functional groups of the amino acids that make up the active site. It brings the chemical groups of the active site into position that enhance catalysis of the reaction. Catalysis in enzymes active site. The end substrate is typically held in the enzyme's active site by weak bonds, such as hydrogen bonds. The conversion of a substrate to product happens rapidly, and the product is released from the active site. Because enzymes emerge from reactions in their original form, small amounts can have huge metabolic impacts. Enzymes will use a variety of mechanisms to lower activation energy. For example, substrates may be oriented to facilitate the reaction. Substrates may be stretched to make the bonds easier to break. The active site may provide a microenvironment that favors the reaction. And amino acids in the active site may participate in the reaction. So you can get different ways in order to accomplish the lowering of the activation energy. The rate of an enzyme catalyzed reaction can be sped up by increasing the substrate concentration. When all enzyme molecules have their active sites engaged, the enzyme is saturated. If the enzyme is saturated, the reaction rate can only be sped up by adding more enzyme. Enzyme activity can also be affected by some local factors such as temperature and pH. It can also be affected by chemicals that specifically influence the enzyme. Effective temperature and pH. Each enzyme has an optimal temperature at which it catalyzes a reaction at maximum possible rate. Up to this point, the reaction rate increases with increasing temperature. Beyond this point, the rate of the reaction will begin to drop. Enzymes begin to denature at temperatures beyond their optimum because what's happening is the enzyme as a protein will start to unfold. The optimal temperature of an enzyme is dependent on the environment in which it typically functions. For example, the optimal temperature for human enzymes is about 37 degrees Celsius, whereas the optimal temperature for thermophilic bacteria is about 75 degrees Celsius. Each enzyme has an optimal pH that is dependent on the environment, and most of these optimal pHs are slightly acidic to more neutral. However, if you go to an optimal pH that's too acidic or too basic, you can end up denaturing the enzyme as well. Enzymes can have cofactors. These are non-protein helpers that bind to the enzyme permanently and reversibly with the substrate. Inorganic cofactors can include metal atoms such as zinc, iron, copper. Organic cofactors are called coenzymes, but again, they're non-protein components. Most vitamins either act as coenzymes or provide other raw materials needed to make them. You can also have enzyme inhibitors, certain chemicals that will selectively inhibit the action of specific enzymes. If an inhibitor forms a covalent bond with the enzyme, then the inhibition is usually irreversible. Many inhibitors bind to enzyme by weak interactions, resulting in reversible inhibition. There are two types of inhibitors. You have competitive inhibitors, which will bind for the same active site as the substrate. Non-competitive inhibitors will bind to another site, an allosteric site on the enzyme, not the active site, But by the inhibitor binding to the allosteric site, what happens is it changes the shape of the protein. And because you change the shape of the protein or the enzyme, the active site that was originally there is no longer the same shape, which, going back to our lock and key analogy, no longer will fit the substrate. Enzymes within the cell are sort of put into compartments to help bring order to metabolic pathways. In some cases, enzymes for several steps in a metabolic pathway form a multi-enzyme complex. And some enzymes have fixed locations and act as structural components of particular membranes. In eukaryotic cells, some enzymes resolve within specific organelles. For example, enzymes for the second and third stages of cellular respiration are located in the mitochondria. The lysosome, another structure we talked about in the tour of the cell, contains digestive enzymes. They are located within that particular structure. So enzymes can be placed into these various types of compartments in order to catalyze specific reactions. And that is it for introduction to metabolism. Next week, we will have cellular respiration, which is one of the major metabolic pathways that occur in most cells. And that will be chapter 9, I believe.